Started then. My name is Micah Johnson. I am a fourth-year PhD student in the Holtman Lab at the University of Iowa. So today I'm going to talk a little bit about my work on migraine, early life stress, and dysfunctional brain circuitry. So to start out, we're facing a problem in the migraine field when it comes to medication. We have some great treatment options, but not all of them work for everyone. So a major focus of our lab is using a brain-wide network-based approach where we focus on dysfunctional brain circuits to identify potential new avenues for therapeutics. So we know migraine involves complex brain circuitry, resulting in a variety of symptoms that we're just now starting to understand. We know that these symptoms present differently across individuals, and some pharmacological treatments work better than others. We also know from imaging studies that multiple brain regions are engaged during migraine attacks. And so our lab is broadly trying to understand how we can look at the coordinated activity of multiple brain regions at the same time to identify circuits that are regulated properly to develop new therapeutic targets that we can repair and improve outcomes in migraine patients. So how are we doing this? First, we induce a migraine-like headache, or we think is a migraine-like headache in rodents using calcitonin gene-related peptide, or CGRP, which is a peptide that's heavily implicated in migraine. So many of you are probably familiar with CGRP. We build our own electrodes with individual wires that can target up to 15 brain regions at the same time. And then we implant these electrodes into rodents to measure their electrical brain activity. And the electrical brain activity that we're measuring is referred to as local field potentials, or LFPs, that provide important signals which tell us what's happening in the brain, similar to EEG in humans. We can then break these raw LFP signals into different components that give us meaningful information. And briefly, we can look at the amplitude of the signal, which correlates with the amount of activity coming from a single brain region. We can look at coherence, which represents how well LFP signals from two different brain regions are synchronized, or the crosstalk between two brain areas. And then after we find the area of greatest coherence, we can then introduce a time shift to this area, which gives us the directionality and therefore tells us the direction of information flow across two brain regions based on which region is clearly leading that of the other. And moving on to my data, so I'm only showing a portion of what I showed at SFN and what we've collected for the sake of time, but we implanted way more brain regions than this, if anyone's interested. So interestingly, we found that the amygdala leads the thalamus, which is shown here by this peak in magnitude within a low frequency band. So it's about 2 to 7 hertz. And thus we hypothesize that this is where we might expect changes in other LFP measures to occur. And so when we looked at one area of the thalamus, the posterior thalamus, we found increased power. So that's indicated by this darker red signal. After we injected CGRP into our mice, which indicates increased brain activity during while the mice are experiencing what we call a migraine-like attack. But I wanted to figure out a way in which I can observe brain changes a little bit more endogenously. So I use early life stress, which Gretchen Tiagen and colleagues have repeatedly demonstrated that experience of early life stress or early adverse experiences predisposes humans to migraine in adulthood. So to mimic that relationship in mice, I use maternal separation, where I separated pups from their mothers for 3 hours each day, and I limited their enrichment for the first few weeks of life. Then I tested their migraine-like behavior in adulthood. I don't have time to explain all of these behavioral tests, but to briefly summarize the results, I saw an increase in anxiety-like behavior, photophobia, which is light sensitivity, and then also mechanical allodynia, which is pain to touch that isn't normally painful, in my animals that underwent early life stress and now were treated with CGRP. So my big picture goal here is to be able to do this early life stress paradigm in my animals and record their brain activity, compare it to what I'm already seeing in animals that we're inducing the migraines in, and then determine what brain connections might explain this increased susceptibility to migraine in adulthood after experiencing early life stress. This is particularly important because early life stress is something that most people don't really have much control over. So I ask, what if we can identify where the brain circuitry is dysregulated in this population, repair these dysfunctional brain circuits, and therefore improve outcomes for migraine patients despite experiencing stress that's out of their control. And so to conclude, let's take the stress out of migraine treatment and repair dysfunctional brain circuitry to improve the outcomes of migraine patients. Thank you for your time.
All right, great. Thank you so much.